this question, we have a diagram showing a part of DNA molecule, right? Okay, and then we look at the key. They're showing us that this is the hydrogen bonds. So revision, okay, DNA, right? We know that DNA is a double helical structure, okay, double helix. Okay, it's double because double means it has two strands. Okay, now not only that, we need to know that the two strands are actually in anti-parallel to one another. Okay, they are anti-parallel to one another. Okay, one is from 5 to 3, the other one is 3 to 5. Now how do we know 5 prime, 3 prime? We look at the number of the carbons, the orientation of the carbons here. So we know that this is carbon 1, 2, 3, 4, 5. So this is 5 prime, this is 3 prime. So this is 3 to 5 prime in that direction, and 3 to 5 prime in this direction, anti-parallel. So the DNA is a double helix structure with double stranded, meaning it's two strands. Okay, make sure you remember that one. And make sure that you know, okay, the subunits of a DNA are nucleotides. Okay, so identify structures E and F in this case. So E. Okay, E is the sugar, so make sure you know what sugar, okay, from the definition here, okay, deoxyribonucleic acid. Okay, I hope everybody remembers the full name of DNA by now. See, deoxyribonucleic acid. Okay, right, that means the sugar, E, that's the sugar. Okay, the sugar would be deoxyribose. Okay. Okay, and then structure F requires us to identify the base. Okay. Alright, so for the base F that they wanted us to identify, the answer is cytosine. Okay, how do we know that? Okay, revision time. Okay, the hint that they are giving us here is that if you can see here, we have 3 hydrogen bonds. Okay, now 3 hydrogen bonds, okay, are only formed between, okay, the bases cytosine and guanine. Right, 2 hydrogen bonds are formed between adenine and thymine. Alright, so we already see that adenine here forms 2 hydrogen bonds. Okay, this is going to be thymine. Okay, and then 3 hydrogen bonds, it's either cytosine or guanine. How do we know that this is for sure cytosine and not guanine? Now next you need to remember the rules of the description of descriptions of purines and pyrimidine. Now in class, I asked you to remember about Qt pi, right? Yes, pi, okay, that refers to pyrimidines. Okay, now what is the definition, the difference between purines and pyrimidines? Okay, so please remember, pyrimidines only have single ring structure. Okay, whereas purines have double ring structure. Okay, now from Qt pi, you should already see that the 3 bases that belongs to pyrimidines are cytosine, uracil, and thymine. Alright, so now we're down to, okay, deciding whether, so how do we know that F is actually cytosine? Okay, because here it has a single ring structure, right? Okay, single ring structure, that's a single ring structure. So single ring structure, it has to be, okay, a purine. Alright, and according to the complementary base pairing rule, okay, a purine will form a bond with pyrimidine. Okay, since we know that purines bonds with pyrimidines, so from the first pair here, we have adenine. Adenine binds with thymine. Okay, we know for sure because then we have this 2 hydrogen bonds. At the same time, look, adenine is a purine, thymine is a pyrimidine. Okay, see, pyrimidine, thymine. All right. Okay, and we know this rule, 2 hydrogen bonds. Next, we have 3 hydrogen bonds. So it means that we only have either cytosine or guanine. All right. So it cannot be guanine, right? Because guanine is a purine. Okay, a purine has a single. So F cannot be guanine because, right, here it has a single ring, right? So single ring can only be cytosine or uracil or thymine, right? In this case, okay, it can only be cytosine. Okay. Instead, the next one here, that can be guanine because it has a double ring structure here. So because this is a purine, this is a pyrimidine. Okay, this is single ring, this is the 2 ring structure. Okay, draw a circle around one nucleotide. So this is how we can draw the circle. So this is one nucleotide. Why? Because a nucleotide will always have, okay, phosphate group, okay, in this case ribose, okay, one deoxyribose, sugar, right, and then the nucleotide base. So that is the definition of a nucleotide. Also not nucleotide, nitrogenous base. Okay, there you go. Name the covalent bond that links 2 nucleotides together. Okay, you should know by now this would be the phosphodiester bond. Okay, where is that phosphodiester bond found? Here you go. That's the phosphodiester bond, right? Because it links between one nucleotide, this is one nucleotide, to another nucleotide, so this one, right? So that's where the phosphodiester bond is. Okay, which then forms what we also call as the sugar phosphate backbone. Okay, sugar phosphate backbone, same. Okay. Figure 6.2 shows RNA-based sequences on the short length of primary transcript, and complete by writing the DNA-based sequences of template strand used to form the primary transcript. So basically, okay, you follow the complementary base pairing rules. Okay, in this case, quite simple. So C, A. Remember, this is the primary transcript. We must write the RNA base. RNA only has adenine instead of thymine. Okay, adenine with uracil. Okay, so continue. C, G, A. Okay, T, A, and then G, A, T. Next, in eukaryotic cells, what happened there? Okay, in eukaryotic cell, okay, the primary transcript is modified to form mRNA. Explain how primary transcript is modified to form mRNA. Alright, so this is under mRNA modifications, the different ways of how the mRNA is going to be spliced, meaning it's going to be cut. Okay, what are we cutting? Okay, so mRNA splicing occurs where the introns are removed, okay, okay, introns. This is the non-coding parts of the DNA, and the remaining exons are joined together. Okay, so that's the coding parts of the DNA. Alright. Alright, so a good note that you should be aware of, that when it comes to mRNA splicing, okay, same gene, okay, can give us different types of proteins. Okay, why? Because of the way how the mRNA is being spliced. Okay, meaning depending on how the same genes, which result in, okay, different types of mRNA, right? So we have same mRNA, sorry, not different types, we have the same mRNA, but depending on this mRNA is being spliced, you can actually form different proteins. So that's what makes it very, very interesting. Now, another way of how we can describe different ways of how the mRNA is spliced is called alternate splicing. So different ways of splicing or cutting the mRNA. Okay, so be aware that interestingly, one gene can lead to different types of protein despite having the same gene producing the same mRNA. Why? Because depending on how the mRNA is being modified, how it is being spliced, meaning which introns and exons are being which introns are being removed and which exons are joined back together, it actually, okay, can be translated as different proteins altogether. Proteins, take note. Okay, the next question is asking about translation. mRNA is translated at the ribosomes to form a polypeptide. Describe how the process of translation results in the formation of polypeptide. Now, several key terms that we want you to take note of is that we want to see mRNA codons, okay, tRNA anticodons. Okay, anticodons, the idea that the anticodons are actually complementary to the mRNA codons. And then how we can relate to the functions of tRNA molecule, okay, carrying specific amino acids, and then as it carries these specific amino acids together close enough that they're able to form peptide bonds, and this, okay, polypeptide process, okay, is continued to join, so more and more amino acids join together in the process called elongation until, okay, a stop codon is reached. So we're going to look at how we're going to be describing translation in this question. All right, so this is how I would write down my answer for 5 points. Okay, let's have a look. tRNA has anticodons. Okay, what is anticodons? Which is basically the codes that are complementary to the mRNA codons. Why is that important? Because, okay, when translation starts, it starts with AUG on the codon, okay, mRNA codon, which codes for methionine. Okay, translation always starts with methionine. Okay, then what happens is that a tRNA molecule will carry specific amino acid. Okay, we've got this one, okay, specific amino acid, okay, which is methionine, to the ribosomes, and then complementary base pairing occurs. Okay, look, okay, where then the next tRNA will carry the next amino acid to the ribosome, okay, carrying the two amino acids together, which will form peptide bond. Okay, and then next, the first tRNA will leave so that the next tRNA will be able to enter. Elongation occur where the length of the polypeptide chain continue to increase sampai bila? Until a stop codon is reached. And see how I wrote the stop codon in capital letter di sana. Okay, so that's how we can describe how translation occurs. Okay, this is the second stage of protein synthesis. Okay.